Hello students, I am VLN Swarupini, faculty in Biochemistry from Sri Venkateswara College of Arts and Sciences affiliated to Adikavi Nannaya University, Rajahmundry. Now I would like to discuss about the topic enzyme specificity. Okay, and we'll see what is enzyme specificity and different types of enzyme specificity. Okay, now let's start the topic. Enzyme specificity is nothing but the tendency of all the enzymes to catalyze a specific site or specific reaction. That means based on the enzyme specificity only, enzyme catalysis or the uh, reaction catalysis takes place. And this tendency is seen between the enzyme and the substrate. Okay, that means enzyme and substrate, they form an enzyme substrate complex, and the based on the specificity only, particular enzyme bind to a substrate. Okay, and every enzyme is completely highly specific in their actions. That means specificity is very high in this enzymes. Okay, due to specificity nature of the enzymes, there are uh, different kinds of enzymes are classified into mainly three types. And these three types are stereospecificity, reaction specificity, and substrate specificity. If you see this, you can clearly understand this. Enzyme specificity. This enzyme specificity is divided into stereospecificity, reaction specificity, substrate specificity. That means specificity is the characteristic exhibited by every enzyme. Okay, and complete thousands of enzymes which are present in our biological systems are classified under these three categories based on their action. Okay, specificity is basically dependent on again stereospecificity, reaction specificity, and substrate specificity. And this substrate specificity is again uh, divided into three types. Those are absolute specificity, relative specificity, and broad specificity. These are the three different types of specificities which are subdivided under the substrate specificity model. Now let's see about stereospecificity. Okay, stereospecificity is one of the important uh, type of specificity exhibited by the enzymes. And the stereospecificity name itself explains it is dependent on the stereoisomers. Okay, this is the kind of specificity exhibited by stereoisomeric components. And stereospecificity can also be called as optical specificity. First of all, what is stereoisomerism? It is nothing but components which have same molecular formula but they differ in their structural configuration. The structural arrangement is different from each other but they share the same molecular formula. Okay, and these stereoisomers which show enzyme specificity is called as stereospecificity. And the enzymes which uh, show this stereospecificity, this uh, show this is shown on only one enzyme. That means the enzyme act only on the one isomer. That means here stereoisomers means two compounds. They have similar molecular formula but different structural configuration. In both of the stereoisomers, enzyme acts only on one component and these exhibit stereospecificity. Okay, and it also explains simply three different regions of substrate molecule. That means this substrate molecule consists of three different regions, and these three different regions are completely complementary to the surface present on the enzyme. Okay, that means these three different regions present on the substrate molecule have complementary to the surface of the enzyme, and this substrate bind to the surface of the enzyme, and these isomers exhibit stereospecificity, and this is due to the interconversion of the reactions. Here if you see an example, L-amino oxidases act on L-amino acids, whereas D-amino oxidases act on D-amino acids. If L-amino acid should be converted into its complementary aldoses or ketosis forms, then this L-amino acid oxidases acts upon them. And if D-amino acids should be converted into their complementary uh, D ketosis or D aldoses, then D-amino acid oxidases react upon these particular components. Okay, and the next model is reaction specificity. This reaction specificity, by the name itself we can tell that this specificity is shown based on the type of reaction. Okay, for every reaction a specialized enzyme is involved in catalysis of the reaction. Okay, and the same substrate can undergo different types of reactions, and different enzymes will be required for that particular different kinds of reactions. That means one single component may undergo different reactions. Okay, that means for example, just see the reaction, you get, you know, you get an idea. Here, take oxaloacetic acid as an example. This oxaloacetic acid undergoes different reactions like reduction, decarboxylation, or transamination and acylation. Okay, here each individual reaction, that means reduction, decarboxylation, transamination and acylation, each reaction require different enzyme, even though it is a single substrate. To when it participates in different kinds of reactions, it requires different kinds of enzymes. So that is what reaction specificity. Based on the reaction specificity, the substrate requires different kinds of enzymes. Okay, and the next type of specificity is substrate specificity. That means here specificity is mostly seen based on the substrate. That means what kind of substrate is being involved in the catalysis, enzyme catalysis, based on that specificity will be present. And these are again three types: absolute substrate specificity, relative substrate specificity, and broad substrate specificity. Now let's see what is absolute substrate specificity. Okay, what is absolute substrate specificity? The enzymes which act on a particular substrate are classified under this absolute substrate specificity. That means nothing but any enzyme which act on single substrate. That means this enzyme is unique for that particular substrate only. This kind of enzymes are classified under this absolute substrate specificity. Okay, example here is glucokinase enzyme. We all know this glucokinase enzyme is the first enzyme that we see in the glycolysis process, which converts glucose into glucose 6-phosphate. See the reaction. This glucose is being converted into glucose 6-phosphate by the enzyme glucokinase. And also enzyme urease. This is also a absolute substrate specificity enzyme. This enzyme urease converts urea into ammonia by elimination of one carbon molecule. Okay, these are the two examples for this absolute substrate specificity. And the next type is relative substrate specificity. This relative substrate specificity is nothing but enzymes which act on structurally related substrates. That means substrates which are structurally similar on that particular substrates. If an enzyme acts, this is categorized under relative substrate specificity, and this is generally depend on specific group. That means the group which is specially present on that particular structurally related components, and simply we can say they act on a specific group or on a specific bond. For example, glycosidases, these are the enzymes which act on glycosidic bonds of carbohydrates. Okay, as we all know, carbohydrates consist of glycosidic bonds, and to break or to act on these glycosidic bonds we use glycosidases enzymes, and these are can be categorized under relative substrate specificity. Why? Because this doesn't act on one particular carbohydrate. These act on all carbohydrates which consist of these glycosidic bonds. Okay, so structurally all carbohydrates are structurally similar to each other. Okay, and glycosidic bond arrangement is uh, similar in the carbohydrates. So as these are structurally similar components and the enzyme glycosidases acts on them, these are categorized under relative substrate specificity. An example you can see trypsin and chymotrypsin. Okay, these are the two examples for relative substrate specificity. And also oxaloacetic acid can undergo the reactions like reduction, decarboxylation, transamination and acylation. So this can also be one of the example. And see, lipases are the enzymes which cleave the ester bonds of lipids. See here, this is the general reaction of lipids. Okay, CH2O, C double bond O, R1, CH2O, C double bond R2, CH3O, C double bond R3. This is the general reaction of lipids, and this is on this lipid lipase enzyme acts and it forms the ester of this particular lipid component, and this is also categorized under this uh, relative substrate specificity. Okay, and trypsin hydrolyzes peptide linkages which are present in the amino acid arginine, and chymotrypsin cleaves peptide bonds attached to the aromatic amino acid. So these are the few examples for the relative substrate specificity. Now let's see about broad substrate specificity. Some enzymes acts on closely related substrates. Okay, and these are called as broad substrate specificity enzymes. And don't get confused between the substrate relative substrate specificity and broad substrate specificity. In relative substrate specificity, structurally related substrate enzymes act on structurally related substrates. Here closely related, that means here there is no resemblance of structure between the substrates, but in relative substrate specificity we see the action of enzyme on structurally related substrates. Okay, don't get confused between these two categories. Okay, and here the Best example is hexokinase. This particular enzyme acts on wide variety of carbohydrates. If you see the reaction, okay, this is the example. If you see this, you can completely understand. Hexokinase acts on all these carbohydrates like glucose, fructose, mannose, galactose, and these carbohydrates are further converted into their uh, complementary glucose 6 phosphate or fructose 6 phosphate, mannose 6 phosphate or galactose 6 phosphate. Okay, so this means all all these are not these are closely related substrates, and on all these substrates this enzyme hexokinase acts, and these are further converted into their end products. Okay, this is about enzyme specificity. Okay, hope you got all the topic. Thank you, students.